Hi everyone, the room will start in around 10 minutes, so we still have some time to join, like some time until we start, but welcome everyone. And yeah, we will have a talk from Dr. Bennett from Oxford University, who will share a new treatment target for chronic pain. Hi Dennis, hi Hart, Dean, and Nora. Hi Wisdom, do you want to come up or just listen? I'll just invite you. Welcome. This will be an. How are you today?
Morning, Katerina. Doing okay. Hey, was Hi, Katie.
Hey.
Hi Katie, do you want to join us on stage? Welcome. Hi Nora, you're new to Clubhouse, I heard. I see. How are you? Can you hear us?
Yeah, I can hear you, Katrina.
Welcome. Today we'll have a talk about new treatment target for chronic pain from a professor from Oxford University in UK. So yeah, we'll start in around 10 minutes, everyone, and yeah, we'll be listening to his talk, and then we can ask questions in the end. So yeah, it will be a very interesting room, and I'm very interested because I know actually a few people that have chronic pain issues, and yeah, it's it's very important that we come up with better medications. So let's see. Hi, Katie. How are you doing?
Hi, love. I am okay. How are you? I'm excited for this conversation. I'm not gonna lie, it's 2 a.m. in Australia for me, so I don't know how long I will hang around, but I'll definitely listen to the replay. And oh, time zones are so challenging with these talks, but very exciting conversation. And thank you so much because it's a topic that I'm sure many people would love to hear about.
Yeah, well, thank you for coming at 2 a.m. And yeah, I totally understand. Yeah, the time. I'm glad that we have recordings. I have a few people emailing me after, after they saw what they missed, that I should send the recording. So all the time. So yeah, it's challenging to get the time, especially between Europe and like European time zone and then Asia, Australia. It's very challenging to get to get it lined up. It's almost impossible. So um yeah, but gladly we have the recording. So hi everyone, we'll start in around 7 minutes. Thank you for coming, and yeah, it will be a really interesting room. So feel free to hang out with us a little bit.
Yes, and thank you again. It's Katie. Thank you, Katerina, for always organizing these amazing conversations with amazing scientists. And I will definitely, and I encourage everyone else to listen to replays and share the room. And as I said, I'm going to listen in, and when I am going to sleep, I'll mute myself to the audience. But I'm very excited for this conversation. And I really encourage everyone to invite your friends to the room. Many people in the world with their different experiences are experiencing chronic pain. So, such a beautiful, amazing topic that I'd love to hear more about. Love you.
Yeah, thank you, Katie. That's so nice of you. Yeah, I agree. It's a very important topic. A lot of people are suffering from chronic pain, and we don't really have good medicine that doesn't at the same time create more problems such as addiction. So yeah, this is really important. I want to support this type of work as much as I can, and in our little way we're supporting to make awareness what's out there, and hopefully yeah, we make a lot of progress in these fields that are so important. So thanks everyone for being here to support, and yeah, we'll start in a few minutes. Hi Natalie, hi LT, and Jenny, Mardi, Yazar, Susan, Shank, Lance. Thank you for coming. We'll start in around 4 minutes. So, in the meantime, feel free to check out the paper link that's pinned on top of the room to read up if you are interested in the meantime. And yeah, we'll start soon. Thank you. Hey, Dr. Mariam, do you want to join us on stage? Welcome. How are you today?
Hey guys, I'm good, thanks. How are you all?
Good, good. Thank you. Thanks for coming.
Yeah, luckily it's good timing today.
Yeah, we just spoke about, the different time zones and how it's so hard to create a schedule that works for everyone. It's almost impossible. But we cover now a few different time zones, usually every week. So I hope everyone gets a chance to be here while it's happening once in a while. So I'm glad it
Yeah,
worked out today. Hi, Jamie. How are you today?
Hi, Katerina. I'm very well, thank you. How's your day going so far?
Good, good. Thank you. Oh, and there's Dave. How are you today? Thank you for coming.
Yeah, I'm good, thank you. Right, I'm not used to this format, but I seem to be in, which is great.
Yes, you are in. We can hear you. Everything is perfect.
So you can.
Pleasure to meet you, Dr.
Brilliant.
Bennett.
Okay. Yeah, nice to
Welcome.
meet you all.
Yeah, thank you for, making this clubhouse
So,
account
Katerina,
and joining. It's very nice of you. Thank you. Please go ahead. I'm sorry.
do you want me to? So, if I'm going to talk through this work, do you want me to share? Is there a way of sharing slides or showing slides, or I can just talk it through verbally? What do people want?
Yes. Do you want to? So we have to share them as a link. Do you want to
Okay.
send them to me really quick? And while
Okay.
we have I'll introduce you to the crowd, and in the
Yeah, sure.
meantime, I'll I'll upload it and make it into a link. So yeah, it just takes me a couple of minutes. So
Yeah, no worries. I'm
sure.
sending.
Hi, it's Katie. I just wanted to say welcome, Dr. Bennett. We have, if you can see at the top of the room, welcome to Clubhouse, firstly, and thank you so much for coming, being a part of the Science Society, and thank you, Katerina, for inviting you as well. If you can see, if you go to the top of the screen, there is a link if you can, like, we can all click on that. Everyone that is in this space, what we call a room, can click on that, which is a link to your paper. But if you do have a slideshow or PowerPoint, as Katarina said, you can send it to her, and we can also link to that as well, and we can follow through that way. But we just love to hear your science and ask questions, and what an amazing opportunity to speak to such a respected scientist as yourself. Thank you.
That's a pleasure. Katerina, I sent you the slides. Did you get them, or I can just start on the paper?
I'm waiting, but it's fine. I'll introduce you, and then usually Jamie will ask like a couple of general questions, and in those minutes, I guess the email will arrive, and I'll.
Yeah, sure. And if not, I'll just click on the link and we can talk. I'll just talk you verbally through the paper, and the
Okay.
figures will work just fine. So yeah, let's start on that basis.
Okay, perfect. So welcome everyone to the Science Society, and of course a special thanks to you, Dave. And let me introduce you to our guest speaker. Before we start, Dr. David Bennett. He received his Bachelor in Science at the United Medical and Dental School, Guy's and Thomas, and he did his PhD in the Department of Physiology, the United Medical and Dental Schools of Guy's and St. Thomas, and his MBBS in Pathology, Obstetrics, and Gynecology, Clinical Pharmacology, Medicine, and Surgery, also at the United Medical and Dental Schools of Guy's and Thomas. And his areas of interest are preventing peripheral nervous system injury and promoting its repair. So Dr. Bennett is focusing on understanding how the peripheral nervous system responds to injury, and to promote effective repair and prevent the development of adverse effects such as neuropathic pain. And he uses many different techniques to study it. And he's also a member of the London Pain Consortium, and he leads Dolor Risk at Horizon Initiative, and that has 11 participating centers which are investigating the risk factors and determinants of neuropathic pain. And he was awarded an honorary SCO professorship at the University of Aarhus in 2019. Yeah, and currently he's a professor of neurology and neurobiology and head of the division of clinical neurology at the Nuffield Department of Clinical Neuroscience at the University of Oxford. So yeah, we are very honored having you here today. And yeah, as I said, if it's okay with you, Jamie will ask you a couple of questions, and then and then we get into your science. Thank you.
Sounds good.
Thank you, Doctor. That is a really impressive range of topics that you've managed to cover. Wow, that's quite humbling to hear that. First of all, in the Science Society, we welcome you officially. And what we like to know is, it's become a favourite topic, a personal favourite of mine, when we ask you, where did you first get an interest in science and at what time in your life? And if you have any stories like that you would wish to share with us, because we always find that incredibly engaging, and let us know the person behind the science. What got you into science in the first place?
Right. Okay. Yeah. So it sounds a bit dull, but I think I was always interested in science from a very young age, and just always enjoyed it at school. I was given a chemistry set at about the age of 9, which caused kind of some chaos in my house when I spilled various chemicals that then caused terrible stains on my carpet that my mum complained about. So I always liked doing experiments, and I always liked kind of the idea of testing new vistas of knowledge, which I thought science was all about. I kind of, I suppose biographically, I kind of couldn't decide whether I wanted to do a pure science degree or medicine. Um, Interestingly, I kind of did a bit of work experience, and from my work experience, I got sent to a really, really dull lab that was testing water around the UK for bacteria, and that kind of put me off science a bit actually, because it seemed very repetitive and process driven and not at all creative. So I ended up doing medicine, partly because I liked the people side of medicine. And then, actually, kind of the way I really got back into experimental science was slightly accidental. I in the UK, medics often do an intercalated kind of specialist science degree in the middle of their medical degree, and I did one in neuroscience. And I worked with a couple of very, I'm sure you hear this story a lot, very inspirational mentors, Steve McMahon and Pat Wall. And I kind of really enjoyed kind of actually doing proper lab neuroscience at that time. And then actually, it was pure chance. So Steve McMahon was, who was kind of supervising that science work, was meant to go and work in Genentech. This is kind of in the early 90s, to do a project there in San Francisco. His wife became pregnant, and she was like, "I don't want you going to San Francisco for the summer." So he said to me, "Why don't you go in my place, and you can kind of go and work at Genentech for a couple of months? It will be a good way to spend your summer." And that's what I did, and I had a fantastic time working in Genentech, which was doing some very exciting work on neurotrophins and growth factor receptors. That got me hooked on experimental science. And when I came back to medical school, said we want to set up an MB PhD scheme. How about we fund you to do a PhD? And that's exactly what I did. I didn't really look back from there. I really enjoyed kind of neuroscience. I did go back and finish medicine, and then I kind of trained as a neurologist, but I always knew I was going to go back and set up my own lab. And that's what I did. So I guess the interest was there from a very early age, and then sparked off by some great mentors.
No, that is actually a great story. You wouldn't actually believe how many times we love hearing that curiosity is what's driven a person, and it can sometimes be this. That's what's actually so remarkable is the simplest things can spark on such a fascination. And with regards to you saying the mentor thing is a story. I don't think we can get tired of hearing about fantastic mentors that have changed people's lives. It's such a great thing. And lastly, so what is it that's got you into this specific field of the paper that you're about to talk with us today? Is there a story behind that? What got you to this point?
I actually, in some ways, in the sense that um, when I was doing that neuroscience degree, back in 1992 or whenever it was, seems like a long time ago now. I really found the lectures on pain very interesting, and the reason I found it interesting was for a number of reasons. Um, the the circuitry around pain is incredibly plastic, and I really found that plasticity very, very interesting. It could be mediated from the terminals of sensory nerves in the skin to the spinal cord to the thalamus to the cortex. So it kind of it seemed to span the whole of the nervous system. And it involved both neuronal cells and non-neuronal cells and inflammatory cells. So I really found it an incredible model system to understand plasticity within the nervous system. That's number one. And then number two, of course, it's incredibly clinically important. So I really like the fact that the basic neurobiology seemed really interesting because it was changed so much. And then also it was a very important clinical problem. So I have to say, I kind of got hooked on pain from my very earliest experiences of neuroscience. And I'm one of those people, I've always been interested in the pain field, and my lab has always had a focus on that. So I have an interest in the topic, and then I kind of have brought various techniques to bear on that interest, rather than being someone that delivers a technique, then kind of applies that to different fields. I've always been interested in the topic of pain and tried to study that in a very multidisciplinary manner.
That's incredible and fascinating, and it actually warms us up quite nicely. Thank you very much for your answers. We really appreciate it. And now, whenever you're ready, just relax and get into your talk, and we're all ears. Thank you.
Okay, great. So, Katrina, I don't know if the slides have come through. If not, it's fine. I'm just going to work from the paper.
I didn't receive it. So if it's okay, good to use the paper. Sorry about that.
No, that's fine. I wonder actually, maybe they're too big and they're getting stuck in NYU or some email server. So let's just work on the paper, and I'll give you the background. Right. So I've said I'm interested in pain, and one of the interesting things about pain is tremendous individual differences, and some of those individual differences will affect all of us in this room. So gender has a big impact on the way you perceive pain, and also genetics has an impact, and that's part not all, but part of that individual variation in how we perceive pain is determined by our genetic makeup. And the data from that is from a number of pieces of evidence. Is partly from traditional twin studies. And if you compare monozygotic and dizygotic twins, you can see that certain aspects of pain are really quite heritable. It does depend on exactly what you're looking at, and that's because pain is not one entity. So obviously, pain is a perception that's triggered could be triggered by a number of different stimuli, whether they're noxious thermal, whether they're noxious mechanical, whether they're chemicals. There are lots of different things that can trigger the sensation of pain. And so the genetics as to how heritable our perception of pain is actually depends on the sensory modality, whether it's thermal, chemical, or mechanical. And so I've been interested in understanding pain, and obviously human genetics is one way you can get insight into that. And we set up a collaboration in Colombia. And actually, again, it was just to give you the history of that, it was a bit accidental as to how this came about. And it was because I was interested in genetic variants in ion channels. And actually, there was a very large family in Colombia where they had a monogenic pain disorder, and that was kind of inherited in an autosomal dominant manner. And these family members that were affected would get very severe truncal pain and triggered by cold and becoming hungry. And it was very poorly understood until, with collaborators in UCL, we did the genetic linkage, and we found linkage to a variant in an ion channel, TRPA1. And that was a story that was published in Neuron back in 2020. But it kind of triggered off this. To study that family, I went to Medellin, which is a very interesting town in Colombia, where we kind of neurologically when we met the family, we tried to understand what was going on in the sensory nervous system and why we were getting their pain state, and we worked out that they had this gain of function variant in TRPA1, and TRPA1 is a ion channel that responds to environmental irritants, both extreme cold, but also irritants such as mustard oil. And this channel was becoming hypersensitive and driving this pain state in this family. And when we went over to Medellin in Colombia and started collaboration both with local scientists in Medellin, but also Andres Ruiz Linares, who's on this current paper, and they were part of a big study which was trying to understand using the fact that in Latin America you have this genetic mixture of Native American populations, immigrant European populations, and also African populations, all admixed. And they were actually part of a big project across all of South America trying to understand the genetic basis of physical attributes. And they were finding that this was quite a powerful genetic approach. And we said, well, look, actually, at the same time as kind of looking at physical attributes such as skin color, well, we could study pain and try and understand more about the genetics of pain. So actually, this kind of collaboration dates right the way back to 2010, where we kind of came to this realization that we could work together to understand more about the genetic basis of pain. And we set up a protocol using quantitative sensory testing. And all that really means is that's a flash word for saying that we give a sensory stimulus and we ask and rate the perception that people perceive in response to that stimulus. But that quantitative sensory testing is helpful because it allows us to quantitate what we're trying to measure and really kind of test the pain circuits to try and get insight into how they're working. And one aspect of that, and that's kind of the topic of this current paper, and kind of comes to figure one of the paper, which is to say that pain is interesting in that many sensations, and for instance, hearing and vision, often if you give a repetitive stimuli, you habituate to it, it becomes less noticeable. Actually, pain is the opposite. If you give repeated stimuli that are noxious, that evoke, stimulate nociceptors, which are those sensory neurons that respond to stimuli that may cause damage, actually the pain perception gets bigger. And the idea of, you can kind of give that the technical term, which is wind up, which is if you give a series of repetitive noxious stimuli, actually the pain perception increases during the course of those stimuli. And that's an example of temporal summation. And figure one is just showing the idea that we're giving a repetitive punctate stimulus to the forearm, and people's pain rating increases. If you compare the rating for the whole train of stimuli in panel B compared to the single stimulus, it goes up, and that's a very significant increase. And then panel C gives you that idea that I was speaking about, is there's this huge individual variation. So although almost all, not all, but almost all people do wind up in the sense that they have more pain perception during the course of stimuli, you can see the degree of that wind up varies massively. Some people, it's just a small increase in pain perception. For some people, they're what we call their wind up ratio, which is the ratio for the pain perception, as it were, during the total train of stimuli versus the singular stimulus, may be 4. So that's a massive increase in pain perception. And you could then use modern genetics to harness and try and understand what the basis of that wind up is. And that's exactly what we did. So we managed to, so in this paper, we're kind of publishing the data from almost 1,000 participants, but actually I can tell you we've gone on and done, I've repeated this on almost 2,000 now with the same result, which is that we ran a genome-wide association study. So those that don't know what that means, what we're doing is we're looking at single nucleotide polymorphisms across the whole of the genome. So in panel D, you've got this thing called a Manhattan plot. We're looking across the whole genome, so all the chromosomes here, which are on the x-axis, and then the y-axis is significance. And we're trying to see, are there particular single nucleotide polymorphisms that associate with our trait of interest? And in this case, that trait is wind up as a measure of temporal summation. And you can see in kind of chromosome 14, there's this line of dots that go up above the red line. The red line is kind of the line we use for genome-wide association. And that is then shown in more detail in panel E, which is showing that kind of line of single nucleotide polymorphisms that are associating with the wind-up ratio are all in a gene, or very close to a gene, called SLC8A3. And that was of immediate interest to us. And that is because SLC8A3 is a gene that encodes for a sodium-calcium exchanger, sodium-calcium exchanger 3. And that's shown in a locus zoom, what's called a locus zoom plot in panel E. And then in panel F, it's showing kind of a dose response, in the sense that the more that you have of the minor allele, that's the rarer allele, the higher the wind-up ratio is. And then I guess the other thing, just to say verbally, that's kind of not kind of specified on this figure, but I think is an important point, comes back to kind of what I was saying about where we started on the genetics. And that's the fact that the most associated single nucleotide polymorphism, it's the one in purple, starting with RS1159, is actually pretty much only present in African populations. So if we'd have gone and done traditionally what's often done, which is a genome-wide association study just on Caucasian populations, because we've got less genetic variety in those populations, we would have completely missed this signal. So it's kind of, and I should also say that we do see overall that ethnicity does play a part in the way we perceive pain. So this gave us this initial genetic signal, which we thought, kind of this was a great finding when we had it, because it immediately seemed to give us insight into biology. And I should say we then went on and we started with this group of almost 1,000. We then repeated that on a completely separate group, which also recruited in the same way, and we found exactly the same signal, which was very reassuring. And then we worked with Luda Diachenko, who's based in Montreal, and she had a cohort where they'd done wind up in a slightly different way in North America. But if you look at gene level, again, we were finding a nice association at gene level between this gene and the wind up ratio. So this human genetics was a great starting place, and then we wanted to kind of delve down into biology. Now, if you look at the kind of both, I guess, publicly available data sets, whether they're for human or for rodent, we could see that NCX3 was expressed, the sodium calcium exchanger was expressed within the nervous system. And we could then look at that in more detail, which is what we did next. So if you've got your PDF open, if you go to figure 2, we're now trying to say, okay, so sodium calcium exchanger 3, where is it expressed in the nervous system? And we were immediately interested in the spinal cord. And the reason kind of we put a lot of focus on the spinal cord in this paper is that there's pre-existing data, actually data from one of my mentors, Pat Wall. He had shown that an important part where one kind of important correlate within the nervous system of where this summation of response to noxious stimuli happens, where this wind up is taking place, is in the spinal cord. So we thought there's pre-existing data on electrophysiology of the spinal cord. Let's see, is NCX3 expressed there? And the answer was yes, it is expressed there. So we looked both kind of panel A, just looking generally at neurons, and we can see the red signal is the RNA scope signal for NCX3. We can see that it's expressed within dorsal horn neurons. We can then try and say which type of dorsal horn neurons, and actually the answer to that is VGLUT2 is marking excitatory interneurons, GAD67 is marking GABAergic inhibitory interneurons, GlyT2 is marking glycinergic inhibitory interneurons, and it's expressed in all of those populations. The expression, which is shown in panel E and kind of F, is kind of quantifying that. It's somewhat more highly expressed in the deeper laminae of the dorsal horn of the spinal cord. And we know that the dorsal horn of the spinal cord is a key part of processing of nociceptive information. We can see, as I've just said, it's in all of those 3 different interneuronal populations. And the other thing that we did is if you think about it, what happens is if you think about the nociceptive pathway, you have nociceptors, they're carrying information about tissue injury from targets such as the skin to the spinal cord. They terminate within the spinal cord. There's then complex circuitry within the spinal cord that processes that information. That involves lots of these different interneuronal populations. And then you have projection neurons that transmit that information up to higher brain centers, such as the parabrachial nucleus and the thalamus. And you then get, ultimately they will project to the cortex, and you will get the perception of pain if you've got sufficient activation of that pathway. So an important output of the spinal cord is projection neurons, and we know that FOX2A is a good marker of projection neurons. And we could see that NCX3 is also expressed within those FOX2A projection neurons, again, when we're doing double labeling with NCX3 and FOX2A, and you can see that in panels H, I, J, and K. So this kind of data was reassuring to show us that the gene that we're interested in is expressed within the spinal cord, and indeed it's expressed within lots of interneuronal populations and also projection neurons. I should say, I'm not saying that this gene is only expressed within the spinal cord. We did see some expression within sensory neuron populations as well, and for reasons of time I won't go into that now, but we can discuss that. I don't think that expression in sensory neurons is modulating this temporal summation, the wind up that I'm talking about. It's not where the locus of activity is, but it may be interesting for other reasons. So then moving on to figure 3, we've seen that it's expressed in the spinal cord of mouse. So the question is, does this gene that we identified in a human genetic study, does it have any impact on pain behavior in the mouse? And luckily, André Herschels in Belgium had already created mice which don't express functional NCX3. And so in the panels in figure 3, there's kind of usually three comparators. There's a wild type, there's a mouse with a heterozygous mutant that will be haploinsufficient for this gene, or homozygous mutant will be virtually no functional NCX3. And then we can look at pain-related behavior, and we can do that in a number of different ways. As I said, there's different modalities. So we can use pinprick to look at mechanical sensation. We can do von Frey, which is also a measure of mechanosensation, and then look at thermal sensitivity of these mice with Hargreaves or with various hot plates. And we're really looking to see how the animals respond to these various stimuli. And the bottom line is that didn't change. And maybe that's not such a surprise, because the in the human genetics, this variant in NCX3 only affected temporal summation. It didn't relate to any of the other. We had done lots of other different measures of pain in the humans, and they didn't relate to these variants in NCX3 at all. It was just the wind-up ratio. So in a way, the mouse behavior is kind of mirroring that. Motor behavior also wasn't affected. Now, the formalin test is a test whereby you have two phases. So if you look at panel G, again, we've got these three different groups, and it measures the animal's response in terms of shaking and licking to injection of formalin in the paw. And there's an acute phase, which happens in the first 10 minutes, and then a second phase, which happens kind of in the next kind of 20 minute to 60 minutes. And the acute phase is mainly thought to be just the acute activation of nociceptors, those sensory neurons that respond to the formalin as it's injected into the skin. And the second phase, there's quite a lot of pharmacological data that involves processing at a spinal level. And what was interesting to us is that in animals that either are haploinsufficient or completely lack NCX3, you can see from this graph that they're showing much more pain-related behavior. And so, again, that is something of a mirror of the human data, is that we know that sensitization, what we call central sensitization at the level of the spinal cord, is important from previous data. And actually, we're seeing that indeed it is enhanced in a dose-dependent fashion with NCX3. That's just quantified in panels G to I. We also looked at the pain-related behavior in the mouse after a neuropathic pain injury. And again, in the absence of NCX3, it was this pain-related behavior was enhanced after in a nerve injury model, and that is shown in panel J. So this kind of, I guess, produces nice validation in a mouse that what we've identified in human genetics does seem to be valid in terms of a target that affects pain behavior. And then we wanted to understand the mechanisms. And now if you move to figure 4, so sodium calcium exchanger, the clue is in the title. What this exchanger tends to do is in the forward direction. If there's a lot of calcium within a neuron, it will export that calcium. It has a relatively high threshold, and it uses the sodium gradient to do that. So it's important for the homeostasis of calcium within the neuron. That's the general forward direction mode of this exchanger. There can be some subtleties, is that if you were to massively sodium load a neuron, then actually it may start working in the reverse direction. But let's stick with the forward direction for now. And what we found is when we cultured dorsal horn neurons from mice lacking NCX3 versus the wild type, as I've said, it's a calcium exporter. And you can see if you lack NCX3, the baseline calcium is higher, and response to a depolarizing stimulus, which will normally raise calcium within the cell, is enhanced. And you can see that kind of there's some quantification here with kind of responses in a number of different ways. Not only look at the peak, but you can look at the area under the curve, but also you can look at the, as it were, the rate of decay. And the rate of decay slows down, as well as the peaks and the area under the curve being higher. And essentially what it's showing is kind of fits with what we would expect in the sense of the rate of removal of calcium from the neuron is slowed down in the absence of functional NCX3. And I guess that's interesting because people hadn't really, obviously people had thought that sodium calcium exchanges may be important, but they hadn't really thought about this in relation to the dynamics of calcium responses within dorsal horn neurons of the spinal cord. And so this kind of was nice proof of concept in vitro that this is important in determining calcium responses in dorsal horn neurons. Something that the reviewers asked us to do, and actually it was a good question. It's something we've been trying to do for a while, and I have to say that these experiments, which are going to be shown in Figure 5, were really delayed by COVID because we've been trying to set this up for actually a couple of years with my collaborator Steve McMahon at King's, and it had been really tricky getting the mice to them, and also just setting these up because of various COVID stops and starts. And actually, I guess just in a bit of remembrance. So Steve McMahon was one of those mentors I was telling you about. He's a very good, fantastic pain scientist and a very good mentor to me. And he sadly died of cancer whilst we were just completing these experiments. So this will sadly be probably the last paper I ever publish with Steve, but it was kind of a really nice piece of work to work with him on. And Kim Chisholm, who is on this paper and really drove this work, did a fantastic job. So what we wanted to do is say, okay, we've got this data in vitro, what happens in vivo? I'm sure many of you know that the technology for in vivo calcium imaging has massively advanced over the last decade. And Kim nicely back-labeled projection neurons. So as I've said, the final output of the spinal cord will be projection neurons that then project to higher center. So she used a virus to retrogradely label them with expressing GCAMP6, which is a genetically encoded calcium indicator and can tell us about activity within those neurons. And the bottom line is, could we see the same enhanced kind of calcium levels in vivo that we had previously demonstrated in vitro? And in short, the answer to that is yes. And actually, there were some very striking differences if you compare the homozygous mutant mice to the wild type. If you just look at panel B, you can see much stronger calcium response within those projection neurons. And then that is kind of quantified with individual neurons shown in panel C, and then a kind of average response shown in panel D. And you can see if you give repetitive electrical stimuli, you're seeing a much higher peak calcium response and some slowing of the clearing of that calcium. And that is frequency dependent, and that's really just shown in those traces in D, and then the various kind of average responses shown in F to the different frequency of stimulation. There was a feeling as well that the kind of rate of rise before it gets to a plateau was higher in the mice lacking NCX3. So it was nice that we kind of really had a nice correspondence between what we'd seen with the in vivo calcium imaging with what we'd previously shown in vitro. So the kind of next part of this was, obviously calcium imaging is helpful. It will reflect what's going on in terms of neuronal activity, but it's not the same as measuring action potentials. And Tony Dickinson and Ryan Patel have been collaborators for a long time. And we said, okay, so actually wind-up was a term that had originally been coined by Pat Wall, as I've said, who was an electrophysiologist working on dorsal horn, and he had noted that you get this progressive increased response to repetitive electrical stimuli in the dorsal horn. And first of all, we kind of, in figure 6, actually, this is essentially negative data, although it's data that we expected to be negative. And that was to say, if we give in panel A mechanical stimulus, that's a von Frey hair at different strengths, do we see any difference in the number of action potentials when we compare wild type to heterozygotes or homozygotes? And the answer was no, and the kind of real traces are shown in panel B. If we give thermal stimulus, kind of going from kind of pleasant warmth to kind of unpleasantly painful heat, at least in the human, then do we see increased action potential? And the answer was no. And again, that fits with the behavior, because if you remember, the acute behavioral responses to noxious mechanical and thermal stimuli didn't change. And the same with brushing the skin, that's shown in E, or using ethyl chloride to cool the skin, so cool responses, that's shown in G and H. And again, no change in the receptive field. And again, we wouldn't expect the receptive field to get bigger. So the kind of acute responses weren't the same, but the key question is what happens when you give repetitive stimuli to kind of mimic this effect of wind up. And that is shown now in the next figure, which is going to be figure 7. And this now is looking directly at wind up. So this is giving electrical stimulus to activate nociceptors, those sensory neurons that respond to tissue injury, and then measuring action potentials within dorsal horn neurons. And I guess in panel A, you can see it's kind of, you can see quite clearly that if you compare the 16th to the first stimuli, you are getting more action potentials, even in the wild type, although it's a small change in the wild type. But if you compare the heterozygote or homozygote mice, we're seeing much more wind up with a huge number of ongoing action potentials being fired, and much more post-discharge response in the mutant mice. And we can really see very clear wind up, even at a frequency where wind up's pretty poor. And that, I guess that's shown in the graph here, where again, it's quantifying the number of action potentials for each stimulus. And as you can see, there's a slight increase in the wild type, but there's a much bigger increase in these animals that are lacking NCX3. And that's just quantified. And we tried to give repetitive stimuli at a very low frequency, which is 0.2 hertz. That's in the panel in A. And then we could increase the frequency, and that's the panel in B. And again, we're seeing more wind up even at this higher frequency in the mutant, either heterozygote or homozygote mutant mice compared to wild type. So this is kind of a very nice electrophysiological correlate of what we've seen in the human psychophysics and also in the animal behavior. If you give repetitive stimuli, probably because of that failure to clear calcium quickly enough, you're getting a potentiation of the response of dorsal horn neurons, and you're getting more action potentials, and ultimately that is leading to a greater perception of pain. Just to say again, it was a reviewer's comment. It was a good reviewer's comment, and it relates to we'll stay on figure 7, panels E to G. And the reviewer's comment was, okay, so you've shown us that you projection neurons. So what's the kind of final output of the spinal cord, rather than just recording directly from dorsal horn neurons? And we could kind of answer that because you have something called the flexion reflex, which is if you give a noxious stimuli, ultimately you will activate motor neurons, and you can, I guess, see that as one measure of the output of this interneuronal circuit that subserves pain. And we could see again in the mutant mice, that's shown in panel EFG, and again, maybe just look at the traces at the very bottom. You can see this huge potentiation of the flexion reflex, so the activity of motor neurons after giving noxious stimuli in the NCX3 homozygote mutant mice. So the output, even although we may get increased activity of both inhibitory and excitatory interneurons, overall, this is leading to more excitability within these dorsal horn circuits. And then the end kind of figure, which is in figure 8, was to say, well, can we exploit this? We've shown that NCX3 is important for pain signaling within the spinal cord. And we've shown that if you have less NCX3, the spinal cord is hyperexcitable and you see more temporal summation. So what about if we then increase NCX3 levels? And the way we did that is we used a adeno-associated virus to express NCX3, and we used an isoform of NCX3 that's particularly highly expressed in the nervous system, in brain and spinal cord, and that's NCX3B. And we could then inject that into the spinal cord. So we just selectively increase NCX3 expression within spinal cord neurons. And we found that we've got nice expression, as we would have expected. We weren't getting any change in acute pain behavior. And that's because, as you remember, we want to be looking at sensitized states. So we went back to that test of a sensitized pain state that I was talking about earlier, the formalin test, where you have the second phase that involves lots of sensitization at the level of the spinal cord. And we could then when we increase the levels of NCX3, so now what's happening is that is shipping more calcium out of the cell. Hopefully, it's reducing that sensitization that happens in the dorsal horn neurons. And in fact, we could see a nice effect on pain behavior, particularly that second phase of the formalin that really relates to spinal sensitization. So what that tells you is actually this is an interesting treatment target that's kind of gone all the way from basic human genetics now to testing efficacy in animal models. I'm not saying we would be injecting a virus expressing NCX3 into humans. Ideally, what you might want to do is to see if you can find small molecules, for instance, that could increase the activity of this sodium calcium exchanger. So yeah, it's kind of human genetics doesn't always work out in such a linear story of kind of going down to a target that you can test in a mouse. But this was one example that was kind of really fun to work on, and for me, a great collaboration. And I hope I've emphasized that here between lots of friends and collaborators in Colombia, but also actually across Europe, and particularly with King's College London and UCL, and then with a great student at Oxford called Theodora, who kind of led all the work as part of her PhD that she did with me in Oxford. So happy to take any questions.
Yeah, thank you so much for this really great presentation and for your work. Katie, you go first. She's in Australia. It's 2 a.m., but she's too excited about the talk of yours. So please, Katie, go ahead first so you can go to sleep.
Thank you so much, Katerina. Hello, it's Katie. Dr. Bennett, what a fascinating conversation. I said at the beginning I was going to go to bed, but I just couldn't keep away. It was keeping up with your paper and your amazing talk. So, just very briefly, I'm a geneticist. However, my work is usually in sharks. However, I've worked with the human genetics lab, and I have some health conditions personally that have drawn me into human genetics a lot more. And interestingly, one of the topics that I saw a specialist over the past few weeks, and he was talking about the interaction with spinal or trauma injuries to the neck or spine, and how that might interact with different genes. I can, certainly send you those or put them in the chat, those genes, but it sounds very relevant to my situation and many others around the world. How, as you said, with the effect of the spine and how that can interact with certain genes. I would love to hear a little bit more about that. I understand you're working on mice models right now. Again, I can give you the I don't want to swear, but Dennis knows we call it the it's MTHF. Yeah. Yeah. I'm sure, David, do you know, Dr. Bennett, do you know the gene I'm talking about? It sounds like the mother effer gene. But the interaction, no, but it's very interesting because the doctor and researchers that I am working with are finding a correlation with a gene or several genes, not just that one, genes, and different dysautonomia, different autoimmune diseases, and the correlation with also neck and back injuries. So it sounds so relevant. I'm going to stop rambling. As I said, please forgive me. It's very late in the morning for me. Thank you.
No, thank you. And yeah, I think there's going to be multiple genes that relate to pain, and some of those will definitely, their locus of activity will be the spinal cord. I think traditionally the human genetics of pain has been quite challenging, but partly actually just I mean, because if you really want to look at, say, common genetic variants, you need really quite large cohorts of participants, and also the phenotyping for pain needs to be done really, really carefully. So I think it's kind of interesting that I would say pain genetics is probably a bit behind many other fields, and that's because it's so challenging. But yeah, I completely agree. There's going to be multiple genes at the level of the spinal cord that will be interacting with spinal pain. And Katie, yeah, you definitely get the prize for staying up the latest, I think.
Yes, I agree. Thank you. I have two questions. One, I would like to ask for our friend that is not here that represents glia, and then so those that factor is also expressed in glia. I'm not sure in which regions, like if because glia vary a lot depending on where they grow. Basically, do you think there is also that glia also involved, and maybe are you planning or collaborating and looking into the glia involvement?
So, I mean, I think glia undoubtedly have an important role in chronic pain, and there's lots of interest in neuropathic pain where people have shown direct evidence of, for instance, microglia, particularly in males, actually sensitizing this whole system. There's also data that astrocytes have an important role in chronic pain as well. Um, in our hands, with NTX3, we really see expression is much higher in neurons than glia. I'm not saying there's zero expression in glia, but it's far more highly expressed in neurons than it is in glia in the spinal cord. And in the peripheral nervous system, again, we see it in neurons, we see it in sensory neurons, but we really see very little expression within glia. I guess there may be other brain regions where there is some expression in glia, but I have not done a kind of comprehensive analysis of the whole central nervous system. In the regions involved in pain processing, we really just see it within neurons. There are other isoforms of sodium calcium exchanger, and they might, so for instance, NTX1 tends to be more ubiquitous. So that again might be another avenue to look at.
Yeah, thank you. And my question was, do you see big differences between, or are there any differences between male and female? I'm asking as not a guest speaker here a few, like maybe a couple months ago, that looked at KCC, NKCC receptor
Yeah.
involvement, and there was like a big difference between male and female. So is this pathway basically independent from hormones, maybe?
Sure. So, I mean, I think it's a really important point for pain research, and I think we've all learned in the field that there can be important sex-specific effects on pain. And I mentioned one of them earlier, which is that microglia seem to have a much bigger role of neuropathic pain in males than they do in females mice. For this data, we did do some comparison, and we see the same in the human genetics. We see the same effect in males and females, so it's not kind of sex specific at a human genetic level. And in the mice, we see very similar responses in male and female mice. So for this particular target, we don't see sex specific effects, but it's a really good point. Pretty much in all, whenever you're looking in pain, you need to kind of look at some of the gender sex differences.
That's really interesting. And do you see, so if there are huge individual differences and calcium kind of lingers longer, is there any like a comorbidity of epilepsy, or maybe higher or lower, prone to have migraines, ADHD? Is there any
Yeah,
very pain specific?
that's interesting. So in I mean, in the humans, you've got to remember is what you're doing with human genetics is you're looking at quite subtle effects. So where the SNPs that we saw were intronic SNPs that is probably having very subtle effects on expression. This is not like, so in a way with the mouse, we're taking a kind of extreme example where we're kind of completely ablating that gene. In the mice, actually, we don't see any evidence of epilepsy, but it's a good question. You would worry about hyperexcitability at other levels. People have looked a bit in the CNS of the mice, and there are some effects on learning memory and LTP. So I do think it's likely to be impacting on, in the absence of NTX3, it's likely to be impacting on other populations for that reason. I think you're right in the sense it probably does have effects on excitability in other neuronal populations, and that would be a risk of trying to modulate this pathway as a kind of therapy. That there would be a risk that you might get CNS side effects, and we see that with other treatments that are used for pain. So you just need to bear that in mind.
Unless you use the robots that we had the guest speaker on Monday. We had the guest speaker from Japan. He used micro robots. Like, he invented micro robots that you can activate and deactivate with different types of lights. So you can make them.
Oh wow!
Yeah, it's so cool. So you can make them. And they are trying now to see if the delivery works in cells too. So with one type of light, you make them pack up any type of stuff that you want. Like, they just use tiny plastics. And then with the UV light, you can tell them, okay, now release it. And so you could do that very specific, especially in the spine. Could use
That
those.
does sound cool.
Maybe you can collaborate with him and try it in the mice. I can send you his contact. He's really nice. Anyways, I'll want to give the stage to everyone else to ask questions. I saw Dr. Mariam and Mike, and then everyone else, go ahead. Thank you.
Hey, Katerina. Hey, Dr. Bennett. Thank you so much for such a great talk. It's nice to have you over. So I
Pleasure.
had a few questions, but while you and Katerina have been talking, she's actually covered some of my questions, and you actually inadvertently answered some of them. So I wanted to go back to when you first did your linkage studies. I think you said that was in Colombia.
Yeah.
Um, so one of my first questions was when you were measuring for pain thresholds in different people, how were you controlling for that? And how were you actually measuring it?
So I guess one thing to say is for this particular measure, it's not a pain threshold in the sense that we're giving a stimulus that we know is painful, and then we repeat that, and what we're measuring is how the pain perception increases with repetitive stimuli. The It's a good question as kind of how we control, in the sense that we did a lot of training with. We were very lucky in Colombia to be working with Actually, he was trained as a dentist, Luis Aristegueta, uh, who in the end ended up doing quantitative sensory testing on almost 2,000 people. And one of the advantages was that actually he pretty much ended up doing all of the phenotyping himself. So he became incredibly standardized. And we did kind of have sessions where we taught him very carefully how to do this. And then we would monitor the data very carefully. And we would kind of follow the data over time. The other thing that was important was that for our participants, we made sure it was people that were healthy, that they didn't have any conditions that, for instance, it wouldn't have been fair if someone was, say, suffering from fibromyalgia. It would have completely their results would have been very different from people that don't have any comorbid medical conditions. So we had healthy participants that were not on any medication that may change their pain responses. So we did try and think carefully, and we also tended to have younger people, because also your understandably your pain responses will change radically, between the age of 20 and say 70. So what we tended to have, in fact, was really healthy students at the University of Antioquia. That was by far the biggest participant group that we had. We had a quiet room, we had controlled the temperature. So we were really trying to keep everything as standardized as possible, and to use our healthy participants.
That's great, thank you. And then one more question, and that would be, what would the potential like targeted therapies based on this discovery be? Would they kind of be universal for all, or would they be more like patient tailored based on their genotype? If that is of
So
any.
I think so. Really, I think the genetic part was just a lead in to find this target. I don't think there's any evidence that, uh, in fact, you would say from the kind of mouse data where we've just kind of done quite a blunt experiment of either removing NCX3 or increasing level of NCX3, we were seeing efficacy on pain. So I don't think we'll be personalizing our therapy to genotype. I think our prediction would be is it would work on pretty much anyone in theory. There is always this thing is that, if what conditions might we find it to be particularly helpful in. Well, we've shown on the animal models that it seemed to be efficacious in a model of inflammatory pain and a model of neuropathic pain. So I guess those were things to be think about in human populations. I think one advantage of it, kind of potentially, and it's obviously early days, but potentially, modulating this gene as a therapy, is that, as I've kind of been at kind of pains to point out, it doesn't seem to change your acute pain thresholds. It really seems to be important in sensitized pain. Of course, that's good because pain has a protective function in healthy people. It's just where it becomes chronic persistent pain that is problematic. So it may not be that we want to get rid of pain completely in people. We want to dial it down so that it becomes more tolerable.
That's great. Thank you so much, Dr. Bennett. Yeah, that's my. I'm done with my questions. If anyone else wants to go, thank you.
A pleasure.
Hi, Doctor. It's me again. Thank you so much for that incredible talk, and you're managing to answer a lot of my questions I had for you as well, and your terrific answers. One thing I was wondering is more of a general thing. When you're talking about pain, it was just making me think. Is there a way at all that you can measure, like, how intense a pain thing is neurologically, like how intense the stimuli is, the signal is going up the nervous system, and then is there a way for you to kind of see how different people respond to that? Like I'm more thinking of people's pain thresholds. Some people can seem to tolerate a lot more pain than other people.
Yeah.
And in that, I was also wondering as well, as a side thing as well, is there any kind of direct correlation or supposed direct correlation with pain to damage? Like a cut should only cause a small amount of pain, but it malfunctions if it's a lot of pain, and so on. Thank you very much.
So I think one of the things that's interesting about pain is ultimately it's a kind of subjective experience, and although we can say, for instance, report of how much pain we're experiencing in terms of, say, a numerical rating scale, it's still very personal and subjective. Having said that, I guess some of the techniques used in quantum sensory testing is you're giving a very well-defined stimulus. So you can kind of have a lot of control on the stimulus that you give, and people will then report what they perceive in response to that stimulus. People have kind of tried to come up with ways to get more objective evidence of what's going on in the, as it were, the pain pathway. And you can, for instance, it's difficult, but you can put very small electrodes with microneurography into people's nerves and measure activity in nociceptors. I guess one of the kind of very popular ways of looking at the brain has been functional brain imaging, and that's had a huge impact on the pain field. And broadly, there will be a relationship between the pain that someone reports and the activity in brain imaging across multiple kind of brain regions that are involved in nociceptive processing. I mean, one of the interesting things is there's not kind of one center for pain in the brain. It's very much a distributed cortical network. Some areas more involved in sensory discriminative pathways, some in more the kind of more effective, as it were, the real hurt that lies behind pain. So that gives us more information about the processing of nociceptive information in the brain. I still think ultimately brain imaging has not replaced self-report pain. It's still a very much a subjective experience. I think that's quite a long answer to quite a complex question, Jamie. I hope it answers it.
No, no, thank you. Yeah, it's a really difficult thing, right? That was what I was thinking about. Was it's so difficult to know how much is too much, and it's different for different people. But the other part of that was, is there? Do you have a standard like model in your sort of workings or whatever that says the signal should be so strong versus so much damage, and then anything
So I
outside
think
of that?
one thing is, yeah, I mean, in these experiments on healthy volunteers, we're not causing damage, by the way. So we might be giving a mechanical probe, but actually with our ethics and everything, we're never going to cause skin damage. And the same with the temperature stimuli that we give, they evoke pain, but they're way below what you would need to cause, for instance, a burn.
Okay.
So we're not causing damage to people, and the one thing though that is interesting and is well known in the pain field, if you start looking at clinical pain states, although there may be, the relationship between tissue injury and pain, again, in the same way as your basic pain thresholds vary between people, that also varies massively between people and also depends on context. There's lots of stories of people, say, in very stressful situations in war, losing a leg and saying it's not painful. So it will vary between people and it will massively vary on context. And again, that's what makes pain interesting. And I haven't had time to talk about it, but there's a whole the brain in a way selects what signals are coming up from the spinal cord. So depending on your mood, how well you slept, your attentional pathways, your basic brain circuitry, actually there's a what's called a descending pain modulation system that has a powerful effect on the spinal cord. So there's no simple relationship between the information coming in and the pain that you perceive. It's an incredibly complex but also interesting circuit.
Thank you very much. And no, yeah, I wasn't meaning that you were causing damage. I was more thinking of pain in general. Thank you very much for your answer, Doctor.
Yeah, no problem.
Yeah, does anyone else has questions? Him, Gilbert, Dennis, Victoria, go ahead if you have a question.
I mean, a lot of my questions have already been asked, so that's great. Also, I think I heard you mention that you had a hard stop, Katerina, so I didn't want to push the time too much, but yeah, I definitely in general satisfied with the questions that have been asked. So thank you everyone for asking such wonderful questions.
All right, Dr. Bennett. What did you have to go? I think I remember you said that you kind of have to stop at an hour, so I want to check in with you.
I probably need to go soon because my kids are just getting back from school. So I could take one more question. Otherwise, I am going to shoot off soon. If there's a burning question that anyone has, now is the time to answer it. Ask it.
Okay, yeah. So my question is, are there maybe drugs that are already targeting the specific pathway? Are you developing maybe one in the future? What's next, basically?
Yeah, so good question. So there's no drugs targeting this molecule at the moment, and one of the problems, and I'm not sure I have the solution, is that in terms of small molecules, it's very hard to target NCX3 without targeting the other sodium calcium exchanges. Although it's theoretically possible with structural modeling, it's quite challenging, and that will be something we'll kind of be looking at the future, because really what you would want is a selective activator of NCX3. Other opportunities would be to kind of take a kind of more gene therapy approach, and again, that's something that we're thinking about. And in the pain field, we are seeing, gene therapy is making progress, and kind of things I wouldn't have thought possible 10 years ago are now becoming possible. So again, that's something that we'll be looking at in the future as well.
Well, great. I wish you all the funding and the best of luck for developing this, because I think it's very important and a lot of people are waiting for big breakthroughs in this field. So, it's very important. So, yeah, thank you so much for coming and sharing your work and answering our questions. We really appreciate it. And yeah, maybe if you have some updates, you would like to come back and share some more with us again. This was a great honor to have you here. Thank you.
It's a pleasure, Katerina. Thank you. Thank you for the invite. It's been nice to meet you all. Thanks for the thoughtful questions. It's been fun to talk about the work, and I wish you luck with the platform, which seems to work very well. So thank you.
Yeah, thanks. Yeah, we have been having really interesting talks from all over the world in all kinds of fields.
Great.
Biology to neuroscience to like more direct medical stuff. And yeah, it has been great. So thank you for being part of this journey. So we really appreciate it. And thank
It's
you
a pleasure.
everyone. Yeah. And thank you everyone for coming, asking questions, and for being interested. If you like discussions like these, join the club, then Science Society. And we will have tonight at 9 p.m. EST, Dr. Congreve coming from Stanford University. He will talk about this cool new technology of 3D printing with light converting nanoparticles. So that will be a really cool tech room. And then tomorrow we have Dr. Santos. She is offspring from James Tour Lab. She did this work in his lab, and she worked on light activated antibacterial molecular machines. So it's tiny molecular robots that can target bacteria that are antibiotic resistant, and that's a really interesting work. And then next week, I'm kind of on vacation, so there will be less going on, but we will still have a room on Tuesday about climate fuel burning room, and how different gas levels are changing in the atmosphere. This is focused on helium levels. And then on Wednesday, July 6th, we'll have Dr. Maldonado talking about food addiction, vulnerabilities, and microRNA signatures. And Dr. Spontek will come back. I don't know if you remember him. He was here like a couple of months ago. He will talk about self-disinfecting anionic polymers that attacks all kinds of germs. And actually, Delta Airlines started actually using his very recent research already to make our flights safer during COVID time. So it will be really interesting. So yeah, that's a preview. Thank you, everyone. Thank you, David. It was a great pleasure and honor. And enjoy the rest of your day, morning, and sleep well, Katie. Bye, everyone.
See you all. Bye. Thank you.
Bye.
Thank you, Doctor. Thank you, everyone.
Thank you so
Bye.
much.
Thanks. Bye.